I'm Faith Dickerson, and I'm here with Robert Yolken. We're presenting our findings on longitudinal studies of inflammatory markers in mania and bipolar disorder depression. Good afternoon. I'm pleased to speak with you about our studies on longitudinal studies of inflammatory markers in mania and bipolar disorder depression. This is the outline of our talk. There are going to be two speakers, myself and Dr. Yolken. I'm going to be talking initially about our longitudinal mania study, presenting findings about gliadin. Then Dr. Yolken will be talking about endogenous retroviruses, TNF-alpha, toxoplasma, and MDA antibodies. Then I'll be talking about findings with a combined inflammation score. I'll be telling you about our new probiotics clinical trial for individuals with mania. Then about our longitudinal bipolar disorder depression study with an emphasis on findings about pentraxin 3. By way of background, infectious and inflammatory processes have been identified as potential factors in the pathophysiology of mania. In a previous study from our research group, a cross-sectional study, we found higher levels of C-reactive protein, CRP, in bipolar disorder patients who had some residual mania symptoms compared to bipolar disorder patients who did not have any residual mania symptoms, and also compared to healthy controls. Based on the results of that cross-sectional study, we then did a longitudinal study focused on patients with acute mania. And the goals of this study were to measure the immune markers in these acutely manic patients, to determine changes in these markers over time, and also to compare the markers in the mania patients with markers in our other psychiatric study groups: individuals with schizophrenia, individuals with recent onset psychosis, acutely depressed bipolar disorder patients, individuals with bipolar disorder not selected for mania or depression, and our non-psychiatric controls. The sample for this study included individuals who were admitted to the psychiatric hospital because of symptoms of mania. For most of the individuals in the sample, bloods were drawn at 3 time points: from the day of hospital admission, and these were actually archive samples left over from the medical admission workup, which we were later able to retrieve, from the time that patients consented to our study and were evaluated, which was about 5 days into the hospital stay, and then at a planned 6-month follow-up. Here we see the characteristics of our mania sample. As you can see by the 5th row down, the mean Young Mania Rating Scale score at time of evaluation was close to 19, which indicates significant mania symptoms, even 5 days into the hospital stay. And the majority of patients had a diagnosis of bipolar disorder type 1 manic. Here we see the scores on the Young Mania Rating Scale at time of baseline, that's when they were evaluated in the hospital, and then at the 6-month follow-up. Not surprisingly, for most of the patients, there's a decline in their mania scores, but that's not true for all of the participants. The first result that I will present is focused on gliadin. Gliadin is a glycoprotein derived from the ingestion of gluten from dietary wheat and related grains. Increased levels of antibodies to gliadin develop in gastrointestinal abnormalities, including celiac disease, where the hallmark characteristics are elevated levels of antibodies to tissue transglutaminase and to deamidated gliadin. An association between elevated levels of antibodies to gliadin and schizophrenia were first reported 40 years ago, and there have been some other studies since that time. Studies from our research group more recently have shown elevated levels of antibodies to gliadin in several different psychiatric populations, including individuals with a recent onset of psychosis, those with multi-episode schizophrenia, and bipolar disorder not selected for mania or depression. However, we find that these groups do not have the hallmark characteristics of celiac disease in terms of elevated antibodies to transglutaminase or deamidated gliadin. Talking about these antibodies raises the possibility of an intervention, and that antibodies to gliadin will decrease following dietary limitation of wheat and other gluten-containing foods. So there's the possibility of a dietary intervention. Here we see the results of our testing of antibodies to gliadin and also ASCA in our mania sample over the 3 time points of the study. As shown on the left-hand side of the slide, we see elevated levels of antibodies to gliadin at time of hospital admission, and then at that evaluation time point, which is about 5 days later, and then a decrease at the time of the 6-month follow-up. For purposes of comparison is our control group, which is the dotted line across the slide and the level of 1. We also measured ASCA, which is another intestinal marker, a measure of intestinal permeability and not affected by diet. And we see a very similar pattern. Again, we see elevations at the time of hospital admission and evaluation, and a decreased level at the time of the 6-month follow-up. We also looked at the association between antibodies to gliadin and rehospitalization in this mania cohort. And I should add that in the United States, in this era, patients are hospitalized for very short periods of time. A patient who is admitted for acute mania will typically only be hospitalized for 1 or 2 weeks. It used to be much longer. Now it's very short. And what that means, of course, is that there's a significant risk that the person will be rehospitalized in a follow-up period because they're discharged so quickly. So we have measured rehospitalization and used readmission as one of our outcome measures. And in our 60 patients with mania, about 1/3 of them were rehospitalized for a mood episode in that 6-month follow-up period. And we found that the levels of antibodies to gliadin at follow-up were associated with whether or not they had been followed up, whether they had been rehospitalized in that 6-month follow-up period. We now have a change of speakers.
Okay, so my background is in infectious diseases and inflammation. That's really where my training is, and I only got involved in working in the psychiatric disorders through the support of the Stanley Medical Research Institute and also from working with Faith. So this has all been new to me. So I try to expose myself to some of the concepts and also to get some of the ideas that I think might fit in. And one of the things I've been interested in for a long time is how infectious agents may change behavior. And they may do it directly, but they also may do it through inflammation. So I'll be talking about both of those. And of the types of infectious agents, one that I know you've been involved with here and also I've worked with is retroviruses. And the reason I think retroviruses are so interesting is because they really exist in 2 forms. They exist in an infectious form, in which case they might be considered to be environmental agents, but they also can integrate into the genome and also be considered as genes. So we're all aware that chronic disorders such as schizophrenia and bipolar disorder and mania are probably involved to some extent in genetic processes and to some extent in environmental factors as well. And in some cases those may be different. But in terms of retroviruses, a retrovirus really could be both, because it also can be a gene and also can be an environmental factor. And I think that's one of the reasons that we're so interested. So the data we're going to talk about today actually are some of our data in terms of antibodies to the endogenous retroviruses. And the endogenous retroviruses are parts of the human genome, but they got into the genome by integration of infectious retroviruses many millions of years ago during our evolution. And these retroviruses bear resemblance, antigenic resemblance, to animal retroviruses. And we use the animal retroviruses as a tool to try to look at human exposure. So it's not that we think that people are exposed to these viruses through the animals, but only that the viruses in our genome actually bear resemblance. So that's the type of work that we're doing. And it allows us then to measure antibodies and do the kinds of studies that we can do in large populations. So I'll present some of the data, again using mostly the mania patients that Dr. Dickerson described. The first set of viruses are actually in feline immunodeficiency virus, which is an infectious virus of cats, but as I said, bears homology to viruses in the human genome. And even in a relatively small sample size, the one that was described, there are clearly elevations in individuals going through acute mania, and then these elevations really come back up more or less to baseline and follow-up. And this is really a theme. It's not true for all the markers, and I'll show there's some that don't follow, but I've been very impressed in these mania studies and also in studies in bipolar depression that you'll hear about a little bit later, that something's going on very acutely. Something's happening when the individuals and the patients are coming in the hospital, but then at follow-up 6 months later, not necessarily when they're necessarily better, but after this acute event, these are largely gone. And that's true for the feline immunodeficiency virus shown here. Another retrovirus, murine leukemia virus, is a different set of antigens, and this pattern is actually a little bit different. This pattern is also increased in acute mania, but stays increased at 6-month follow-up. And we don't actually know the reason for that, because that is different than the individuals with bipolar disorder that are found that are evaluated longitudinally, so there is a difference there. So this is something that's happening acutely, but also persisting, and it probably has something to do with the activation of the virus that's going on. Now, exposure to mouse viruses is somewhat controversial, as you know, in chronic fatigue syndrome. I don't think that's what we're measuring here. We're not actually measuring exposure to a foreign virus, but again, an activation of an endogenous virus. One of the interesting aspects of the endogenous viruses is it also predicts rehospitalization. And you just heard about gliadin as a predictor for rehospitalization. And it turns out antibody to one of the viruses, a primate virus called Mason-Pfizer monkey virus, or MPMV, also is predicted for rehospitalization. Really quite a difference between the two groups. So again, this activation, it's very hard actually to see differences in the patients when they're acute. They're all undergoing mania, and the measures we do really don't correlate very well because they're all so active. But I think if you look in follow-up to what happens to them, that's probably a much better idea of what's going on in terms of infections and inflammation. And I think one of the themes of our talk today, I think, is the importance of longitudinal studies, because if it weren't for a longitudinal study, if we hadn't followed these people up, we'd have none of this particular data. So it's only really because the team at Shepherd-Pratt's been able to do these follow-ups and engage in a longitudinal study, we've been able to do this. And as an infectious disease person, I mainly work in the laboratory, but as I look at the data, I've been extremely impressed in the importance of these kind of longitudinal studies, particularly in mania, but I'm sure it's true for other disorders as well. The data on retroviruses is very consistent with other studies. The one on your right actually shows a study that we just published on antibodies to endogenous retroviruses in individuals with recent onset psychosis. On the one on the left is actually a paper that Professor LeBoyer was involved in, and you've contributed patients here, which are detections of retroviral antigens. And again, you'll see that it's individuals with mood disorders, the bipolar disorder shown, that really have the highest level of activation. And that's actually quite consistent to what we're finding in a totally different method. So we're using one method, this is another method, but they're actually coming out with very similar results, and that's very encouraging. We do occasionally find antibodies to retroviruses in control population, but the level is much lower, as you can see. Now, one other point I want to make, these are Western blots, and if you're familiar with them, but every band actually represents a different antigen. So a virus has about 7 proteins, and the proteins are broken down into many different antigens. And one of the things we're finding is if you look at individuals as individuals and not so much as a group, they all have antibodies to the target, but their antibodies are different. So there's a fair amount of individual variation in which protein someone's reacting to. So this individual has an antibody to this virus, and that individual has an antibody, and that individual, but they're different in the epitopes that they recognize. And I think that's where the genetics comes in, because individuals may be exposed to an agent or exposed to an activation, but the people do vary in terms of what types of antibodies they make. And I'll show that in a little more detail in a minute when we talk about antibodies to the NMDA receptor. Okay, we've also been interested in cytokine measures. We don't do quite as extensive cytokine measures as perhaps other groups do, because I'm concerned about the collection of samples and that they be collected and stored in an organized way, that we measure them in a way that's highly consistent. But some of the measures are actually pretty consistent. And tumor necrosis factor alpha, which you're probably familiar with from your studies, is really one of them. And we focused on this because there's also some genetic information that would support a role of this agent, but it hasn't been extensively studied in mania. So the interesting thing about TNF alpha, again, the levels themselves, you looked at the levels, they actually were fairly variable among the group, but they were highly associated, again, with this rehospitalization. So individuals who wind up being rehospitalized appear to be the group that really have the most of the immune activation. We haven't measured a complete panel of cytokines, but at least the other ones we've measured don't have this association. So there's something different about TNF alpha in this population. Now I want to turn now to another infectious agent, one you're probably familiar with its toxoplasma. And I'm not going to talk about the biology of toxoplasma because I think people are familiar with it. Are people familiar with toxoplasma? Okay. So I think people are familiar with this. And the reason we're interested in toxoplasma, there are a couple things that are very interesting about it. One is that if you're interested in an infectious agent, and we're talking about psychiatric disorders, these are lifelong disorders, right? So we need an infectious agent that is capable of persisting in a lifelong situation in someone who's immunologically normal. And there are very few organisms that fill that bill. Usually you get an infection and you get over it, or if it's a serious infection, you have a serious problem with it. But toxoplasma is one of the small group of agents which really can persist lifelong without much in the way of clinical syndrome. So someone who's infected with toxoplasma as a child, as I presumably was, still carrying this organism lifelong, and we don't know exactly about what happens to people that actually have the organism and where it's persistent. And that's one of the reasons that we're interested in doing this. The other aspect about toxoplasma, it's been shown in a number of studies that toxoplasma is capable of encoding genes, its own genes, which actually change dopamine and NMDA and other neurotransmitters. And they do this as part of their life cycle. So they actually have the ability, the biological ability to alter behavior. So we've done a number of studies of toxoplasma, but the one I want to talk about today is in the mania study. And what we found is actually quite interesting. We found increased levels of antibody to toxoplasma in admission and acute mania and at study entry. And again, at follow-up, at 6 months later, these antibodies are decreased. And this is pretty surprising because we think of antibodies to infectious agents as lasting for a long time. But the thing about these antibodies that's different is these are really IgM class antibodies. And the IgM class antibodies are ones that seem to come early in infection. They usually represent an acute infection, but that's probably not true for toxoplasma. This probably represents a reactivation of the organisms that are already present in someone's brain. It also may represent a reinfection of a different type. That's also a theoretical possibility. We actually also find these elevated in individuals with bipolar disorder. And if you look at people with acute psychosis, that's a little bit different because they have antibodies, but you can see from the blue bar that these are actually IgG class antibodies. And we were just going over data from the group here, and increased IgM levels of antibody toxoplasma are also seen in bipolar disorder in patients from your own study. So it's actually kind of interesting because we're finding very similar data in our group in the United States and the group here. And given all the environmental differences and genetic differences, when we find an infectious agent that's true across an association, it's true across two populations, that makes it quite interesting because that suggests that despite all the differences, these organisms are still playing a role. And I think toxoplasma really fits in with that. There are a lot of therapeutic implications of this because we're in the process of trying to develop better medications for toxoplasma. The problem of toxoplasma is not so much getting rid of the organism when it's replicating, but when it's sitting there by itself, it's very resistant to medications. So we're actually trying to develop drugs that will kill the organism when it's actually in its resting state. And we've just succeeded in finding the first set of medications that can do that. They've only been tested in animals now, in mice, but we're hoping to develop them for human use because they're types of medications that have been used for treating malaria in people, and therefore they're suitable for human use. So we can try to do that as well. And then finally, lastly, I'm going to talk about our antibodies to the NMDA receptor because we know that infectious agents and inflammation can do a lot of things, but we don't know how it does them in a lot of cases. So there's been a lot of interest in trying to measure antibodies to the NMDA receptor, and I know the groups here are interested in that, because this would provide a logical way to try to link an infectious agent and inflammation and also a clinical finding. And just to review the NMDA receptor, it actually has two binding sites, one for glutamine and one for glycine. When both bind, as you can see in the animation, the receptor opens. So it's a very tightly controlled receptor, and it opens only for a short period of time. Now, in terms of antibodies, there are many places the antibodies could function. The antibodies could function at this receptor. They could also function at the binding site. So there are many places for antibodies to actually exert their effect. This is what's known about NMDA receptors in psychiatric disorders. It's been shown in a number of individual cases to be associated with acute mania and also with encephalitis, but previously had not been examined in a large set of individuals with mood disorders. I actually, as a pediatrician, took care of a patient, an adolescent that had acute onset, first actually of psychosis, but also of mania, and had one of these cases of encephalitis. So that actually kind of got me thinking about trying to do this in the psychiatric population. So these are results which have just been published, I think this month or last month. And again, individuals in the acute mania phase have markedly increased levels of antibody. Now, from this point to this point is really only a very short period of time. It's only a few days in a population basis. We see the antibodies are actually starting to fall, and they're actually much lower at follow-up. And you'll see some data in a few minutes suggesting a lowering of inflammatory markers in that period of time as well. Now, individuals with established bipolar disorder really don't have those increased levels of antibodies. So this also seems to be an acute phase marker. Now, I mentioned before talking about individual variation. I think this is important because we all know from our patients that patients are a little bit different. I don't think there are any two patients with mania or with schizophrenia that are exactly the same. And there are genetic differences as well. So one of the places that these differences can come up is in terms of response to an antigen. So we're quite interested in taking an antigen, in this case the NMDA receptor, and breaking it down to all the individual peptides. And there may be really thousands of them in any one protein, and trying to see whether people are the same or different. And by doing this, trying to understand exactly how some of these antibodies are functioning. So this is a fairly new technology, so we're still sort of getting to work on it. But this is actually one of those pairs. This is an individual with mania, both at the enrollment and also at follow-up in the pink and then the pink diamonds and the green dots. And what this is this is the whole protein. And this is the first peptide and second peptide and third peptide and fourth peptide. So that's 1,500 different peptides it's reacting to. And what I wanted to point out is that in enrollment, there's actually reactivity to many of the different peptides, and that's probably what we're picking up. And at follow-up, although we don't find much in terms of the antibody levels to the NR2 peptide, there's still some antibody left, but it's only recognizing one of those epitopes. Now, what is that epitope? So we've actually tried to look and see what that epitope reacts to. Although there are plenty of places that antibody could react, this particular epitope is actually one of the calcium kinase domains, and the antibody is very specific to that particular domain. And that's what we're seeing in this particular patient. In another patient, you may see other ones and controls other ones as well. But by doing this, we can actually try to dissect what an individual person is reacting to. And I'm very optimistic about this particular technology and trying to individualize what we're doing, because this particular person with mania, that's clearly what they were reacting to. And individuals that have other individuals that have these antibodies, individuals with lupus or with other autoimmune disorders, they recognize the NMDA receptor, but not this particular peptide. Individuals with lupus will recognize this peptide, and most of the people with encephalitis will recognize that one. That's why I have those up there. Okay, so I just wanted to make one more point. Sorry. Now the question is, how are the antibodies related to infection? And this is actually very hard to study in humans, and that's why we use animal models. And in the case of toxoplasma, we use a mouse model because there's a lot been done with it. And what we're able to show actually very nicely in animals is that toxoplasma infection clearly leads to an increase in these anti-NMDA antibodies and also in changes in behavior. So some of the changes in behavior may be related to the toxoplasma, but some may be related to the antibodies. We also find this correlation in people, but of course in people it's hard to know cause and effect. Here we can actually study that. So by understanding infections and understanding some of the immune responses, we can try to put them together and try to figure out what's actually going on in terms of changing behavior.
So having presented these various findings so far, we wondered if we combined our antibody markers, would we find even more marked differences with our control group? So what we did is we performed a principal components analysis using the 4 antibody measures, 2-gliadin, the NR2 receptor, toxo-IgM, and the Mason-Pfizer monkey virus. And what we see is even more pronounced differences between our mania group at admission and study entry with our control group. So as you can see on the right-hand side of the slide, using this combined inflammation score, we see elevations in the mania group at admission and study entry and a lower level at the time of the 6-month follow-up. And on the left-hand side of the slide, for purposes of comparison, we see our control group, which is set by definition at 1, and different other of our psychiatric study groups. Individuals with schizophrenia, with recent onset psychosis, and with bipolar disorder not selected for mania or depression. We then wondered, would this combined inflammation score also serve as a good predictor of rehospitalization in the 6-month follow-up period? And again, we find that the combined score is quite predictive of rehospitalization. This is a Kaplan-Meier survival curve, and the top line on the figure represents the individuals who have a lower inflammation score at baseline, and they are less likely to be rehospitalized in the follow-up period. I want to turn now to a clinical trial that we are just starting at my hospital in Baltimore, and the clinical trial is based in part on the findings of our longitudinal mania study. This is a double-blind trial of probiotic microorganisms to prevent relapse, to prevent rehospitalization after an initial admission for mania. By way of background, probiotic microorganisms, which you may be familiar with, are non-pathogenic anaerobic bacteria found in breast milk and some other foods, and which have been shown to modulate the immune response. We've just shown in our data that individuals with mania have elevated levels of food-derived antigens, such as gliadin, and the antibodies are associated with rehospitalization. Therefore, the mechanism of action in this proposed trial is to alter the inflammatory response in the intestinal tract of our mania patients to these potentially harmful antigens. In this clinical trial, we're going to be enrolling 66 patients who are hospitalized for mania. And the study medicine is going to be an add-on. We're going to add it on to their existing medication, so that people in the study will be randomized either to a tablet containing lactobacillus and bifidobacterium in fairly high concentration, or to an identical appearing placebo in addition to their regular medication, and they'll be followed for 6 months. The primary outcome that we're going to study is relapse or rehospitalization, and we're also going to look at new mood episodes, symptom severity, and levels of inflammatory markers. So we'll keep you posted, and hopefully in a few years' time we'll have some results from this study. We anticipate actually recruitment is going to go a little more slowly than for our other studies because of the difficulties of enrolling acutely manic people for a 6-month treatment trial. I want to end by telling you about another study which we are doing, which is underway and partially completed. So I have some interim results to share with you. This is a study of longitudinal bipolar disorder depression. By way of background, we know that depression is the mood state that's the most common and enduring in the course of bipolar disorder, and for many patients is also the most problematic. There are relatively few studies to date that have looked at inflammatory markers and inflammation in acute depression in bipolar disorder. The methods of this bipolar disorder depression study are similar to those in our mania study. What we are doing is enrolling people who are hospitalized with acute depression who have a diagnosis of bipolar disorder type 1. And for these individuals, we are collecting blood samples at 3 time points, again from the day of hospital admission, from those archived blood samples left over from the medical admission workup. Then when we enroll them in our study, which is on average a few days later after they're settled into the hospital, and then at a planned 6-month follow-up. Again, this study is ongoing, and we have some preliminary results. We intend to have a target N of 45, at least by the time we are completed. Here we see the clinical characteristics of the bipolar disorder acute depression group. As you can see by the mean score of the Hamilton Depression Rating Scale, the bottom row of this slide, even several days into the hospital stay, this score is quite high, indicating significant depression. I want to turn now to one finding in particular concerning pentraxin 3. This is a marker that has only become commercially available in the United States quite recently. Pentraxin 3 is a member of the pentraxin superfamily, which also includes CRP. Pentraxin 3 is rapidly produced and released by several cell types, more cell types than are released by CRP, so it's more reactive to inflammation. It also has a shorter half-life than CRP, so changes more rapidly in response to the immune system. It activates the classical pathway of complement activation and facilitates pathogen recognition. And because of these properties that I just mentioned, it's under evaluation as a reliable marker of cardiac and CNS inflammation. What we have found, rather strikingly, is elevated levels of pentraxin 3 in the admission of blood samples from both our mania and our bipolar disorder depression samples. As you can see on the left-hand side of the slide is the mania group, and the right-hand side of the slide, the bipolar disorder depression group. And for both of these groups, we see a marked decrease between the time of admission and the time of study enrollment, which is only a period of several days. So we are very struck by these findings and the sharp decline in the levels of pentraxin 3 in this relatively short time period. Here on this slide, we see the same data and also the 6-month follow-up period. Again, you can see the decrease in levels of pentraxin 3 from time of admission to enrollment in each of the study groups, and then another decrease by the time of the 6-month follow-up, but this is not actually significant, the second decrease. Here we see the same data again as in the previous slide, but also showing other of our comparison groups. So we see the mania group, then the acute depression bipolar disorder group, then our control group, and other of our study populations measuring pentraxin 3, the schizophrenia group, the bipolar disorder group not selected for either mania or depression, and then our recent onset of psychosis group. This is a slide that was shown earlier in the talk, in which we found that antibodies to the NMDA receptor are elevated in mania, but not in bipolar disorder depression. And we want to point out that it's interesting that the findings about pentraxin 3 we find both in acute mania and in acute depression of bipolar disorder, whereas the findings about the NMDA antibody levels seem to be unique to acute mania. And again, in the NMDA antibody level study, we see that in bipolar disorder depression, both at admission and evaluation, these levels are not elevated. So now we're up to the conclusions part of the talk. We've shown that patients with mania have increased levels of immune markers and antibodies to food antigens and to retroviruses. We've shown that the levels of some of these markers are lower 6 months later following the acute manic episode, and that some of the markers predict a rehospitalization. We've shown that patients with bipolar disorder acute depression also have elevations to pentraxin 3, but do not have elevations to the antibodies of to the NR2 peptide of the NMDA receptor. We're performing further analyses with our bipolar disorder acute depression group, and again, we are continuing to recruit patients for that study. So what are the clinical implications of these findings? Well, certainly our data support interventions addressed at lowering levels of markers of inflammation and antibodies to food antigens. This could be done, for example, with medications such as COX-2 inhibitors, statins, other inflammatory agents. Some of these trials are underway. Of course, there's always a concern about side effects, but they're very much of interest. And one could also intervene in terms of measures to decrease antibodies to food antigens by a dietary restriction, which is very challenging to do in any population, or through the use of probiotics, as I explained about the clinical trial that we are starting at our hospital. We would like to thank our colleagues at Sheppard Pratt, Johns Hopkins, Columbia University, and rules-based medicine, now Myriad, and we're very grateful to funding from the Stanley Medical Research Institute. Thank you very much, and we'd be